thanks for the invitation. Sorry for just being here today. I had some teaching duty. So I work in a medical school in the north of Paris, and we work in on infectious disease, but we're also interested in evolution. So I'm no physicist, I'm more an evolutionary biologist. And I want first to present the people who really did the work. So two amazing PhD students from the lab, André for the first part, Harry for the second part, and Philippe co-supervised Harry, and Jimmy helped from some of the statistics. And so here, basically, I'm going to show two stories about epistasis, and that are unpublished, and so it's any comment are good. And please, I may introduce lots of concepts, go fast, way too fast. I have 120 slides or so. Feel free to interrupt. Okay, we can. Okay, we're here. Just it's the last talk, so don't hesitate if anything is unclear. So. So what are we after in life? You know, I have a midlife crisis, okay? And so one of the questions is, you know, what are we after? And so for some people it's easy, you know. For politicians, they're after power, you know. For a soccer player like the French team, the World Cup. Now we have 2 World Cup, okay? Then the businessmen after money, but we, as evolutionary biologists, we can agree on something, that we're after mutation, okay? What we're after is basically mutations, okay? But the thing is that we're just as greedy as the others, and we're not just satisfied with one mutation, we want more mutations. And then comes epistasis. Okay, so as soon as you have 2 mutations, okay, you have the effect of this first single mutant, the other one, and then epistasis refers to the surprise I have when I measure the fitness of the double mutants and compare it to this one I could have expected from the 2 single mutants, the addition of the effects on the log scale. So here, positive surprise. The mutant, the double mutant is fitter than expected. Here, negative surprise, negative epistasis. You know, this is bad news. The fitness is lower. So epistasis can be defined, has been defined over the years, thanks partly to Dan Weinreich, to many different flavors. So you can have no epistasis. You have 2 mutations. You can have negative epistasis, but it's just magnitude. The 2 mutations are still beneficial, but to a lesser extent when fitness is higher. Sign epistasis, here you have 1 mutation that is beneficial in one background, deleterious in another one. Reciprocal sign epistasis. So there's lots of literature on that. And basically, why do we care about epistasis? Because the ultimate goal of genetics is to make a link between genotype and phenotype, or fitness. Okay, we want to understand whether it is for, you know, disease, pathogenic. I work in a med school, so we work on bacteria and trying to understand how those bacteria evolve, and we're going to talk about antibiotic resistance, but also for bioconservation, how species can survive in the face of mutations and low population size. We're interested for the mutation effect in bioengineering, and also in personalized medicine or informed choice. So this is something that affects whole genetics, and always behind it, if we could predict the effect of mutation, we'd be happy. And of course here, epistasis is coming as a trouble. But to do so, to do this, to understand how epistasis is appearing, let's go to a simple thing. So let's assume we have a substrate in a cell. This substrate is going to be consumed by an enzyme. I mean, not consumed, but modified by an enzyme. And you have several other enzymes. And then through metabolism, we're going to turn out to a metabolome. And that, through a complex process that we call physiology, is going to turn out to organismal trait that will define fitness. Of course, these enzymes are encoded by genes, and eventually those genes are also regulated by other genes. Okay, so this is the whole process that will lead, and mutation here, through the whole process, will lead in the end to this organismal fitness. And of course, then also the environment may play a role at every single stage. So this is quite a complex process here. Okay, and so our goal in the lab is trying to understand, can we make any prediction about mutation effects? And basically, the idea is that can we split also epistasis in 2 forms? Some form that on which we cannot predict anything, like microscopic epistasis, to take some words by Michael Desai, Benjamin Good, and Sergei Krasiaminsky. Microscopic epistasis will define our idiosyncratic epistasis, those interactions that are super specific to something that we don't understand much about it. And then what we're interested in is that whether there's some global scale, global epistasis, or macroscopic epistasis, where we could try to make predictions. And so Levi presented experimental evolution, and when we look at something that has been found consistently in experimental evolution, is the fact that there's some form of macroscopic epistasis. So here we summarize that in some essay, but with Alejandro Cauce. So here, basically, if we take many different experiments based on yeast, bacteriophage, E. coli, and you take some clones that are diverse for some reason, they have mutations or they're slightly different clones or whatever, but they have different initial fitness. And then you adapt them to the same environment. What you figure out is that the maladapted tend always, okay, to adapt faster than the ones that are most adapted. And so the underlying idea is that there may be some global trend here about epistasis. It's not a proof, a formal proof of that, but this is something that has been seen consistently. And then people have put the mutation, as Lidvai presented. You can reintroduce the mutation in the ancestral background and compute those effects and see how the different mutation. And what's been consistently found is that if you add several mutations, their effect is going to decrease as your fitness is increasing. Okay? The higher the fitness, the lower the effect of the mutation. So if you put the fitness effect of mutations according to the fitness of the background in which they occur, you find that it's mostly, of course, it's biology. Everything is false in biology, but right at the first order. We survive with that. And basically, in most cases, we find that there's this negative correlation between fitness effect and the fitness of the background in which mutation occur. So the question is like, what are the molecular determinants of this epistasis? Can we go further? And as Lidvai say, I'm a reductionist also, and it's hard to understand the wholesale system, so I'm going to present two examples where we had a very reductionist approach. To make it simple, on the first one, I will just focus on one enzyme. Okay? You have all these complex paths, so I'll focus on here. One enzyme, make all the mutation in the gene, and see how it turns out that there's epistasis. And for that, I'm going to take one quite successful enzyme, which is beta-lactamase TEM1. So this enzyme, actually, if you think about it, it was discovered first in a Greek patient in 1964. And so in E. coli, it was found on a plasmid, this gene, just after the introduction of amoxicillin in 1960. So 4 years after it was found. And now, a few years later, 35% of E. coli cells have this gene. Okay, so you think about few copies in 1960s to 10 to the 19 cells, you can call it a success. I mean, I wish you the same. I mean, not for planet Earth though, but okay. So when we focus on one protein, okay, what could be the source of microscopic and macroscopic epistasis? Well, for microscopic epistasis, maybe you're not biologists, so you haven't read those papers, but whenever you have people making those 3D structure, and they all come up with those stories. Oh, this here, I'm always completely jealous of these works because I never understand what's going on. But here the idea is that they always explain, oh, this happened and this mutation is interacting because this histidine is switching to that position and pushing that stuff away. That's what I would call macroscopic epistasis. There's always a histidine, I don't know why, but it's always just a story like that, okay? And what could be then macroscopic epistasis? So here, thanks to work, pioneer work actually by the Nobel Prize Frances Arnold and Jesse Bloom in 2005, the idea came out that protein stability could be one of the global macroscopic source of epistasis in the proteins. Basically, most of the amino acids within a protein are involved in the structure rather than the active site. So most of the mutation effect can be understood through the action on protein stability rather than the effect on the active site. Okay? And basically, there's a nonlinear mapping if you do, you're here for the biophysics, okay? If you plot, I'm a biologist, so I plotted stability that way, but it should be energy the other way around, okay? But we biologists, we go up adaptive peaks while you physicists sink in energy bottom, energy sinks, okay? So we have slightly different visions. And so here, the idea is that if I have a protein, and this is stability, there's this nonlinear model, that a nonlinear function linking stability to the amount of folded protein. And in the case of an antibiotic-resistant gene, I can directly link the amount of folded protein to the fitness. Okay? And so here, based on that, let's assume I'm creating mutants in that background. You see there's going to be quite a large variance in mutation effects. And if I mutate and stabilize this protein and do again mutation in that background, you see there's much less variance. This is some form of epistasis. Okay? The mutation effects are depending on the background in which they occur. So we did that a few years ago with, during the PhD of Hervé Jacquier. And what we did thousands of mutants in beta-lactamase STEM1, and so in this enzyme, and we could see, so what we measure here is the minimum inhibitory concentration of that enzyme. So what's the minimum inhibitory concentration? Basically, when you are sick and you have an infectious disease, they're going to take the bacteria you have to try to test how sensitive is it to antibiotic. And what they're going to do is that they're going to plate this bacteria at different concentration of antibiotic, and the minimum inhibitory concentration will be the concentration at which you see no more growth. Okay? Meaning that you need to put a concentration at that level of antibiotic to kill the bug. Okay, so for the ancestor, it's around here, that's the wild type value. And you can see among the mutants we had that you had this classical bimodal distribution in which you have a long tail of deleterious mutations that have less. You need much less antibiotic to kill the bug here, so the enzyme is less functioning. And a fraction here that are basically non-functioning. Okay? And then we just mutated that enzyme and added one stabilizing mutation that has been discovered in the mid-90s. And this enzyme, actually, you see just with one stabilizing mutation, and these are the same mutations that are plotted in here, you see that there's much less of an effect of most mutations. Okay, so you can see through here that, there's going to be some epistasis linked to stability.
I think that's far away from the active site.
It has nothing to do with the active site. Actually, this mutation was selected because this gene is quite interesting for TEM1, it's quite interesting for evolutionary biologists because, as I said, it's a big success. But in the mid-90s, one thing happened is that the pharmaceutical industry introduced new beta-lactams, okay, new molecules, and this gene was sensitive to those new molecules. But through a bunch of mutation, it happened to become resistant. So with 5 mutations, it can increase its efficiency. This enzyme can increase its efficiency to those new compounds by 40,000-fold. Okay, and so it was predicted in vitro by Stemmer. He put this gene, did some mutagenesis in the gene, and put the new antibiotics, and he could select the mutation and predict the mutation that would occur later on. Some people say that they emerged later on. Some people say that he introduced them to show that he was right. He put them in nature himself, but okay, that's another story. But then, so basically those mutations were seen, and among those mutations, in the late 90s, one mutation occurred that was M182T. And this one was surprising because it was not at all acting on the active site, but it was just stabilizing the whole cell and making the other mutation that modified the active site more efficient in that background. Okay? So the question then is that, okay, we know it happens, but can we try to see how prevalent is epistasis and how much do we have of this micro versus macro epistasis? So for that, we decide to do large number of double mutants. And so here, we focus from technical reasons. In that case, we focus on just this alpha helix that is not specifically close to the active site, but we'll see that it's not either super far. And we just choose that one because it has 11 amino acids, and that was good enough. That was the largest amount of double mutants. There's like about 19,000 double mutants we could do in here, and that was about the numbers we could work with. So basically, what André did is that he took this library, and then we evolve it through experimental evolution, if you want, in the media with antibiotics. So it's a painful experiment because we don't want the population to grow to high density to destroy the antibiotics so that the concentration will change. So we keep the bacteria at low concentration over 4 days, which means 4 nights, also 3 nights. Okay, so that's quite painful. And then we sequence here. So here, there's a trick is that to follow the genotype, we put some molecular barcodes, and so that we can follow accurately the different genotypes. And so basically, for one barcode, we can follow through time the change in frequency, and theory is predicting that it should be straight lines like this, or if it's dying immediately, we can see also a depletion super fast in the early time point. And you know that a change in frequency can be directly computed as a fitness measurement. Okay, so the regression here is the fitness, is the measure of the log fitness. Okay, so basically, after a bit of filtering, we had 15,000 double mutants for which we had quite precise fitness estimate, up to very precise when they have small effects. So first of all, we can look at the distribution of mutation effects and see that a bunch of them are not doing much here. Then we have a bunch of them. This is the distribution for stop codons that inactivate the function. And so here, a bunch of them are quite inactivating. And I'll be conservative and saying that, you know, when you're below -1 in log fitness, I would say that these are inactivating. Okay, that's, I put a threshold here. For the double mutants, a large fraction of them are killing the protein. So now we can look at, for all the different possibilities of amino acids. So with the trick for at every codon, we have all possible amino acids. And so here you can see that you have, you know, the different amino acids have different fitness effects for the single mutants. You can see a good control is that proline here. So proline is an amino acid that's switching, that making a kink in the, changing the direction, and you cannot have it in an alpha helix, okay? And so here the fact that it's red means that all the mutation to proline are almost killing the stuff. So that's a good control, showing that, okay, it's bad to have a proline here. And then we see that some sites are more permissive to mutation. Those ones, there's like much less deleterious effect, and others are really restrictive and constrained there, where there's not much that can happen here. So this is with all the double mutants that we have, and we can see, so in gray is the missing parts, so we don't have the full data set, okay, and here it's expected that, you know, you cannot have 2 mutations at the same codon. And so here, basically, so let's zoom in some parts, and let's zoom. So what we see here is that the single mutant effects on that, on I-127, T128, and you can have here the effects of the double mutants. This is the one that is found. You see that that's the native state. And you can see that here, whenever you have some red on one and red on the other, the result is red, okay? And meaning that if you inactivate it one way or the other way, the result is going to be red, okay? But overall, the rule is also true that if you go yellow to yellow, it's tend to be yellow. Okay, so at first you see doesn't seem to be much epistasis. Okay, there still seems to be dominance. If you kill the protein, whatever you combine a killing mutation with, it's going to kill the protein. Yet one way to see epistasis is just to add up the effect of log fitness. Okay, so that's the predicted log fitness. I add the effects of the 2 single mutants, and I compute it against the log fitness here, observed log fitness. So here are the ones in which I have 2 very deleterious mutations, inactivating mutation. You can see that for both of them, I'm below my detection threshold somehow. So both of them, this is completely inactivating mutation. So there's no recovery. It could have been possible. Some of the ideas of epistasis is that you have those key lock mechanism where, you know, you could have think, oh, if I mutate that, if I mutate the other one, I would compensate. For a large effect mutation in that setting, we don't find any. But then here, if there was no epistasis, we should expect everybody to be along this line here, okay? And we see that there's lots of deviation, and we can even compute the type of epistasis involved. So we can find a distribution of epistasis with large positive ones, large negative ones, and an overall dominance of negative ones. Great, okay? But any artifact would create a distribution. If any errors in the measurement, I would also have a distribution, and I could talk a lot about an artifact and say, oh, it's more positive. So the point is having a distribution doesn't mean anything, okay? So what we need to do is to have some biological meaning out of it, okay? To see whether I'm the first one to be convinced first, okay? Whether is there any meaning under that distribution. And for that, I'm going to try to present you this graph, okay? I'm going to look at one thing. Before I show you that when people have done experimental evolution, what they found out is that there was a connection between the effect of mutations and the background, the fitness of the background in which they occur. They tend to be this negative correlation, that mutation tend to be beneficial mutation, tend to have a smaller effect when they occur in a feeder background. So it means that basically the idea is that presumably if we measure fitness of the background in which a mutation occurs, maybe that tells us something about that mutation, what's the effect in that background. And so for that, okay, let's assume I have a mutation, the yellow mutation, and it's a deleterious mutation. So I'm going to plot it here in the ancestral background, which has, by definition, initial fitness 0. This mutation has, let's say, I don't know, 15% cost or whatever, okay? So I'm plotting it here. Now, in my data set, I have also this mutation combined with the red mutation. And here, there's no epistasis. You see, it's just the add up. And so now I can plot here this mutation in the red background that is also a deleterious mutation with a 20% cost. So here I find that this mutation has the same effect. I make this plot. Now I have also, with my double mutants, I have also the effect of that mutation in the blue background, which is even lower fitness, okay? And so I can make it. So if there's no epistasis, I expect to observe straight lines. Is that clear for everybody? Okay, no epistasis, there's no context dependency. The mutation has the same effect in all the backgrounds in which I found it. Now there's a few issues here, is that because I'm my threshold of detection, this, when you're below that, you're lethal. I'm not able to say what you are when you're dead. And even worse, you know, when you hear you're back from the dead, I cannot quantify much. You know, if you quantify epistasis, you know, when you survive from the death, you know, I cannot say how good you are compared to dead. You know, that's another issue, okay? So here's one mutant. Well, I can measure that when I can see that mostly, you know, there might be a small trend in the end, but there's not much of an effect of the background, okay? So now I see that for this mutation, so there's not much of an effect. Now for that one, I see that there's quite drastic change. So here, within a few % change in the background, you see that the mutation can come to be almost having, you know, less than 10% effect to having 90% effect, okay? So here you see a lot of context dependency. And I have also some mutations that are like this, okay? So here, two things. First, you know, when I see that and when I see those patterns, this is biology. And it means that my distribution and the measure of fitness I'm doing have some meaning here, okay? There's something, it's not just by chance. If it was just noise in the whole system, I would not expect to see those patterns to be dependent on every single mutation. Then I can try to explain now why the hell do I have those patterns, okay? And so first I can count, and I can count so that among there's 209 mutations, individual mutation that I could do in that alpha helix. Among them, there's about 70 that are unconditionally lethal, meaning that in any background, they inactivate the full function. Cannot get anything. I have about 120 in which I saw this strong positive slope in which, you know, they're deleterious and they have a strong context dependency. I have about only 10 that don't show clear sign of epistasis, and most of them are almost neutral. They have almost no effect at all. And I have about 10 also that show this pattern, the last pattern here, is this, that can be extremely beneficial when they're combined with some deleterious mutations, okay? So there's a massive amount of epistasis, okay? Almost all mutation, no epistasis is more the exception in that context, okay? So now I can try to go back and to try to see, I told you that one of the things that could explain such pattern could be epistasis and stability at the protein level, okay? And so here we can see that we can compute fitness as the fraction of folded protein, you know, according to a simple two-state biophysical model. And I can compute, you know, that's the fraction of folded protein, so the relative fitness will be the ratio of the fraction of folded to the one of the wild type. And I can compute here the log fitness based on that we call G. And the fitness of the double mutants will be, I will assume, additivity at the phenotypic level of the delta G. So that goes back to what Armita presented today, that we may have some stuff that are just additive at the phenotypic level, but I may have a nonlinear mapping from that's this nonlinear fitness function from this stability to fitness. So if I do that model and try to plot it against the same curves as the one we saw presently, the background fitness, here I did that for a stabilizing mutation, a highly destabilizing or a neutral mutation, you see the kind of patterns that we've observed before. And so we can do some fitting here for each mutation, try to compute just one value, this stability effect, and we can find some fits to that, okay? So definitely we improved. This is now, in red, it was the previous distribution of epistasis, and in blue, it's now the deviation I have from that model. So first thing is that, okay, we reduce the variance. We explain much better the data, okay? Specifically, what I like as a biologist, I'm not as quantitative as you guys, I like that I have the qualitative fit, I find back those slopes and so on, so qualitatively I'm happy. But I can see that there's still room for improvement. You know, I'm not explaining everything. I'm far from being good, okay? And then especially there's some mutations that do not fit at all, like this one. This one is a trouble because this one is a mutation that the model is predicting that, you know, you don't see much of context dependency here, but this mutation is deleterious. It has 10% cost. And with the stability model, you know, to have a flat line, you only need to be neutral, okay? And so this is not compatible with the model. And this happened at the site, actually. This mutation is really the mutation that is at the site that is the closest to the active site. And then we can try to play around and say that eventually there's not just stability, there's also something linked to the active site, so we can add another component. We can, I'm going fairly fast on that, but we can try to measure how good we are if we add this other component on every single mutation, and we can find that basically all the mutations that are close, here this is the site that is close to the active site, all the sites, the closer you are to the active site, the less good the fit with stability only is. It's like the closer you have to the active site, the more we need something else to have a better fit, okay? But yet, it's not the only thing. We have also here, you see this panel, this seems clear, but you have also outliers like this. And here, so these are, and here you see also this kind of pattern here in that site where you see those mutations, wow, they tend to, you see something fairly flat and even going on, and those are going up. And when you look, all these mutations are just at the next site, okay? They're just at the next residue. So they're not randomly distributed, all those ones making a straight line, and we can go further and show that actually R12220D is moving from positive to negative charge, and the mutations that are compensating are the ones that are going from negative to positive. So there's appeared to be some conservation of charge here. So we can see also that locally, we might have some, the global stability model is good overall, but locally we may deviate from it for some of those idiosyncratic story. Okay, here we need this charge, or here we need this and that. And so here we can try, it's just one statistical way to see that, is that we can try to see the standard deviation to the model, assume that we have two errors, one error to the model that is based on close residue and one for distant residue, and we can find that the error to the model of stability is much larger, almost twice larger, when you consider residues that are in contact compared to residues that are distant. Meaning that when you close to the active site, when you close together, when residues are close, just the additivity that we assumed on the delta delta G doesn't seem to hold anymore. There seems to be more complex stories. Okay? And so the idea is that now we have a more complex picture, because now we need to understand epistasis not only in terms of the classical epistasis that we measure as deviation from the additivity and fitness, but also measure epistasis as deviation from the global epistasis that we have, like from the stability model. So we need somehow to have, we cannot picture it only as epistasis the way we used to measure based on fitness, but also based on deviation to the stability. And so now we have to, epistasis is becoming more complex, because there may be several phenotypes, so we cannot just take just the stability, because we saw there's also the active site, and so now it's becoming even more fuzzy, okay? So just to conclude on that part, we see that there's pervasive epistasis, but still a lot of it is linked and can be explained through a global pattern of epistasis, which works with the additivity at the phenotypic level, and we'll come back to that in the end. But yet, when residues are in contact, there's more also idiosyncratic epistasis, okay? In residue in contact, we have much less power to predict what's going on there. Yep? No?
Oh, I was just going to say that at some point we did a model which has a folding free energy and binding free energy, so it's two axes.
Exactly.
Yeah, just like this one.
Yeah.
And I wonder how much that would capture. That was still pairwise in the energy space. I mean,
so then, yeah, you have now, and you would have eventually to take into account several phenotypes, and that may each of them have this nonlinear stuff and how they interact with one another. Here, I don't have the power. What Jakub did in his paper in a larger data set in a small protein, he had more power and he made something very nice out of it here. So now it's something where we want to go after. Okay? And so there's maybe not only one trait, but eventually several of key. So stability definitely is going to be one major one. But for some residues, not necessarily all around the protein, but at few sites, there may be other phenotypes that matter. Yep.
So do you think, so for microscopic sort of like charge features, right, do you think that if you very coarsely your protein, right, so instead of looking at individual residues, you group things together and come up with some sort of lattice size of some coarse graining measure?
We could. So the idea is that maybe we have this global phenotype stability, but maybe we have some more local ones, and we can capture them. So there's like Rémi, Monasson, and Simona are trying to do something with Boltzmann machine to try to identify some of those modules eventually. So we're trying to work that out with them on that.
And
Yeah.
then the other thing could be
I'm sorry?
To be a bit more pedantic in the
Yeah.
physics lecture. So you were talking about errors in fitness measurements.
Yeah.
So you have replicates, I suppose, right?
Yeah, yeah, yeah.
You can have an error model.
We can have an error model. The worry is that, so there's two worries. First is that whenever we do replicates, it's really tricky to be in the exact same conditions. It's a nightmare because then, as soon as we put antibiotic, the concentration is going to be different. There's no way we can reproduce exactly the same condition from one day to another. So that's going to add some. And then there's some source of noise. We tried to play around on that, but I still have some doubts about it. I tend to find my estimates of error much lower than what they actually are. I played around for a year in Spain. But it's yeah. So I've been playing with that. But it's we can try to decompose. I tried for a long time to decompose the model with the error, measurement error, the stability deviation from that. But I've not been super successful, to be honest. But any help is welcome.
But the fitness, right? Should
Yeah.
kind of be less sensitive to the.
Still, because of the sensitivity of the antibiotic, we're just at a threshold for a lot of them, and so as you switch, maybe it's not as easy. Okay, so that's for this part. Okay, the second question we wanted to look. So that's one case where we looked at the protein. Okay, and now with Harry Campbell, we decided maybe we should look at another part of this winding path from fitness. And so the idea here was to look at the smallest pathway we could have, smallest metabolic pathway in which we had two genes, and on which we could play on the expression and look how, just this time playing on the expression of two genes, we can understand fitness. Okay, so basically, one of the ideas that's been around in the literature for some time is that metabolic network, now we can build them easily from bioinformatics. Okay, so the subjective hypothesis was that maybe just from the structure here of this network, we could predict epistasis. You know, whether you mutate that gene, you express that over, you could predict this all. So this was super appealing because you say, okay, now just from the computer, I can predict that. And so it's been suggested, but it's never been tested. So we say, okay, let's try it on a very simple system with two genes, and let's try to see what's going on. So we choose the arabinose pathway, which is a well-characterized pathway in which L-arabinose is turned to L-ribulose, 5-phosphate, and further down by other enzymes. So two genes are involved, RA and RB. And so here, one specificity that is quite common, it happens to 15% or 20% of metabolites, is that one of the intermediate is toxic. Okay, L-ribulose 5-phosphate is toxic. So what we did is that we put those two genes under inducible promoters that will allow us to move around the expression, the basal expression of those two genes. And then on the promoters, we mutated 12 bases here on each and did all combination and also used barcodes to do that. And so just to let you know, these are the sites we mutated, that the -10 and -35 boxes. And these are where the repressor binds. And so it's mostly independent of the action of the repressor. There may be one site actually that's telling another story. One of them here, which in the end is the one that it does not fit, may interplay with the inducer expression. Okay? And so basically, let's assume that this is an hypothetical fitness landscape here, where we have the fitness would be here the maximum with this amount of RB expression and this amount of RA expression. And then what we're doing is that this is our initial wild type sequence given the inducer concentration that we use. And now we're going to look at the single mutants in RA, and this is going to give us some points here. We're going to look at the single mutants in RB, this is going to give us that. And all the double mutants will be here. And because here again, I assume that the effect is going to be stable according to this. This is, I'm assuming, you see also additivity on expression in those two stuff. And here I can just change the inducer concentration, and like that I can sample different positions in the landscape, and in the air somehow have a global picture of what's going on in that landscape. So a few tricks. I mean, this is a lot through sequencing. We can follow the barcodes, and because we have many barcodes for each genotype, we can have an internal control about the error rate, and we have very accurate, in that system, even more accurate measurement of fitness. So what do we have first? So let's look at, this is the 12 mutation on one, the 12 wild types, the wild type sequence on the 12 site that we mutated on one of the promoter and the other one in one on the environment. And we can look at the single effect mutants. So here going from blue to red means blue means deleterious, red means beneficial. And so here you can see the effect, and we can look at all the double mutants. And so here you can see that, I mean, we're super good because we managed to create a strain in which most mutations are beneficial, actually. So our strain is so sick that, you know, everything's improving. That's pretty cool. Okay, it's positive. Okay, and so here you can see that, you know, there's definitely a pattern, but you can see here these are mutations that we know from previous other studies that these are mutations that increase the expression of the gene because the promoter is initially fairly good. So most mutation will tend to decrease the intensity of the promoter, but these are mutations that increase its activity, and we see that increasing the expression is bad, okay, in that system. And you see there's some consistency in those data. Now we can look at the two other environments we chose according to inducer concentrations, and you can see that here we go from mostly positive to mutations that are mostly negative to an environment in which it's a mixture. So you can see definitely, depending on where you are in that landscape, mutation have variable effects. So just to go a bit further in this description, between environment and 3, there's some form of correlation. Between this one and this one too, this one is even slightly negative, but you can see still that there's some patterns, okay? Some patterns are behind. So to go further, what we need to do is to go back to epistasis, because characterizing mutation effects and interactions, one way to do that is, as we mentioned earlier, is to do epistasis. And here, to go back, because it's a mixture of two presentations I'm doing, so you have some repetition, but you know that repetition is one key part of teaching. You know, repetition is one key part of teaching. And here you see that you have those mutations, and this is no epistasis, and then this would be positive epistasis, sign epistasis, and you have slightly different possibilities when two mutations are deleterious or when they have mixed effects. And what we can do is that, for instance, just try to understand and quantify those forms of epistasis in those different environments, and you can see that what matters to see here is that there's lots of epistasis, more in some environments than others, but also it does change, you know, according to environment, the fraction of the different forms of epistasis. Now we may try to go further and try to characterize that, look at epistasis between beneficial mutation, between beneficial and deleterious, and so on. And we see we capture some of these variants, but the global picture is that it's still a mess, okay? And so basically we see a lot of epistasis that we have a hard time in making sense of it now, okay? Lots of diversity in epistasis and a high prevalence of epistasis. So to do that, we can try, as we did before, is to try to have this connection between background fitness. I told you that in experimental evolution, we have always this negative or diminishing return regression between, you know, the fitness effect of a new mutation according to the background fitness. So this has been consistent. We saw that it was of some information with our stability model. But here, what we have is something a bit less convincing, I would say, okay? We have this kind of mess where actually the correlation is slightly positive. But when we look at individual mutation, this is a specific mutation. You see that there's still some smoothness in here. It's not random. Okay, so there's still something which tells us that maybe it's worth going and seeing what's going on in there, because it's not just random. Here you have two kind of loops. So for that, we can go back and try again, as we did for the protein, to go back and try to find the relevant phenotypes and the relevant level of mechanistic description that would be good to understand what's going on. So for that, we have this pathway, and where we have those expression of those genes. We have a toxic component here that leads to some product that may be used for growth through physiology. And so first, we can assume that fitness is linked to the flux, because we know that this is the source of the resource for the cells. So here, fitness is going to depend on the flux through that pathway. So we can use metabolic control theory, and it's an old theory that predicts that if you have RA activity and RAB activity, you have this kind of landscape. This kind of landscape is extremely smooth, and you can see that the type of epistasis it produces is not compatible with the type of epistasis we saw, where we saw some sign epistasis, many different forms, okay? So this has been shown to be true in some case a long time ago, but we can see that this is not enough. Now, that's what part we did actively with Philippe, we can try to introduce toxicity, okay? And so here the formula is missing actually. But here we can try to induce this component and introduce that the intermediate is toxic. And so given the toxicity of the intermediate, that's the kind of landscape we expect. Okay, where now in here, rather that you should go up here, but because when you're high, you have a high amount of the toxic compound, fitness is decreased in the high expression pattern. Okay, and this has been also produced theoretically in the early 90s, but never tested. And when we do that, we found a fairly good correlation between the fitness of the observed fitness, and the predicted fitness and the observed one. Okay, fairly good. But then there's another component that we know through synthetic biology, and there's actually lots of recent papers that are really nice about that now, that the cost of expression does also come into the picture. And if we introduce that, this is the type of landscape, that we expect. And now we get an excellent fit between the data and the observed and the data, okay, and the theory. And so here also, even though epistasis is not directly fitted, and it's well known that it's really hard to fit epistasis, the distribution, we have also good fit. So this model, I'm not saying is the best model, I'm just saying that qualitatively, this model is doing a good job here, okay? And so we see what's going on. So just to show off a little bit, you can do that in 3D and show that. And what is interesting is that these are the three different environments, and this is what we see that is interesting is that overshooting is quite frequent, okay? So we see that a lot of the mutants are going over the ridge, okay? They're moving the cell from one side of the ridge, of this ridge, to the other one. Okay, and these are all the double mutants. You see that they're close to the surface, predicted by the model, and this is when we do log 10 activities. Okay, and so what does the model teach us? What does the model teach us? So here first we can see that here this is the top view of the fitness isoclines, and so here the patterns described depending on where we are. Here it's given that we start here. This is the patterns of epistasis, where we see here that here we have some forms of epistasis using the color we had previously, some negative sign epistasis, negative epistasis, positive sign. And we can see that according to the environment, there's very different prediction of where epistasis should be. And you can see that the points are colored according to the epistasis we found for them, and we see that it does match really well the underlying model. So what's embarrassing about that, you can see that even in a simple model like this, epistasis can be of many forms. What is encouraging is that these are big patch, so we can still hope that we can identify those patches in this system. Now, one thing that occurs is that with the model we can reproduce those kind of weird patterns that we observed. And these are due to the fact that some mutation here, because we have a ridge, if we overshoot, we can have one mutation that moves us to the other side, but has almost the same fitness. So we have two mutants that have different expression of the gene, but they have the same fitness. But then when you add another mutation, in that case, there's almost no effect. Okay, that would be that one, while in that case, there's a big gain. Okay, so you can see that some cryptic variation is possible in this metabolic pathway. Okay, you have some, at first hand, those different genotypes have the same fitness, so you could think, well, they're the same. Phenotypically, they should be the same, but actually they're very different. And through the addition of other mutation, you can reveal how different they are. Okay, so this is just what I said. And finally, it's also telling us that I told you about this seductive hypothesis about, oh, great, from just the topology of the network, we could predict everything. And we think that is not enough, actually. We need to take into account some of the physiology, like we need to take into account, protein expression cost and also metabolite toxicity. And here, because there's this highly nonlinear fitness to flux relationship because of toxicity. So we need to add more than just what we could get from flux balance analysis. We will need to add more. These details do matter in the end if we want to capture that. Okay? So to conclude on both systems, what we have is that one thing that is key is that we see that, on both systems we can fairly hold and keep this somehow additivity at the phenotypic level, that when we combine mutation, there's an underlying landscape in which we could add the effects on the phenotypic level. And which is kind of interesting because it means that there's still some stuff that can be done somehow. Okay? There's still some hope. But because of the nonlinearity in the biophysics of physiology, that makes epistasis emerge. Okay? So a lot of the epistasis will emerge from that nonlinear fitting from those underlying phenotypic landscape. But what's encouraging still is that if we know the appropriate knowledge of the molecular detail, we can still predict mutation effect. The real worry is to know what does it mean by appropriate knowledge, okay? So in one case, we see that if we knew, for instance, the stability of the protein, that would be good, because then we could predict that all those destabilizing mutation or stabilizing, we knew that, would be good or bad. We could predict that if we know that. Here, if we know where we are in the landscape, okay, globally, we can predict fairly well the effect of mutations. But how can we know that is open for future research, I would say. Okay, and with that, I'd like to thank again, André, who did all the work on beta-lactamase, Harry, who did the work on the ARA project, Jimmy, who helped on the stats, and Philippe also for that co-supervised with whom we work on with Harry, and all the people from the lab in Bichat. Thank you.